Hi, everyone. Quick note before we get into the podcast. This episode was recorded in late December 2019, and in the conversation, we mentioned a deadline to apply to the first cohort of the Healx Rare Treatment Accelerator. This deadline has now passed, but anyone who's interested in this program can still register their interest on the Healx website. That's healx.io, Healx.io, anytime to receive the latest updates on the program. Thanks and enjoy the show. Welcome to the Genetics Podcast. I'm Patrick, the co-founder and CEO of Sano Genetics, and I'm really excited to have not one, but two great guests today. We have Dr. Bruce Bloom, the Chief Collaboration Officer from Healx, and Dr. Mike Tranfaglia from the FRAXA Research Foundation, which is a nonprofit focused on accelerating research in Fragile X syndrome, which is a rare condition that affects millions of people worldwide, and we're certainly going to learn more about it today. I believe it's one of the most common, if not the most common, cause of intellectual disability in children. So while it's a rare disease, it's on the common side of rare. Healx is a company based in Cambridge, UK that was at one point a startup, but now it's growing very quickly due to their very successful model for drug repurposing in rare disease. So we'll go into this in more detail, but Healx uses a machine learning approach to do something called drug repurposing, which takes medicines that have already proven to be safe and effective treating one disorder and applies them to a new one. So there are a number of advantages to this approach, especially in rare disease. And Mike is the medical director and chief scientific officer of FRAXA. So he and his wife, Katie, actually founded the organization after their son, Andy, was diagnosed with Fragile X. So Mike is a medical doctor by training and specialized in psychiatry. So he not only has the personal connection, but also brings some deep medical expertise to the role. And Mike, as a side note, I also noticed when I was doing my background research that you went to the University of North Carolina for medical school, which is also where I went for my undergraduate degree, doing biology and math. So we have a connection there. Go Tar Heels.
Go Tar Heels.
Okay, so just to get started, Mike, I was wondering if maybe you could bring us back to the founding of FRAXA and the original mission that brought you and your wife, Katie, to actually start the foundation in the first place.
Our son, Andy, was born in 1989, which makes him 30 years old now, and that's scary all by itself. But after he was born, we pretty quickly realized that he had some significant developmental delays, and we had a difficult time getting diagnosed at first, but eventually, when he was 3, he got the diagnosis of Fragile X, basically because he finally saw a developmental pediatrician who had seen other cases of Fragile X. So he literally recognized the look, the actual facial dysmorphisms that occur in Fragile X. So after we got the diagnosis, obviously we started reading up on this thing, and I am a doctor, and I'd heard of Fragile X, but going back to the textbooks, I realized that very few of them had more than a paragraph about Fragile X. It was this sort of mysterious condition that nobody really understood very well. We started to reach out to some of the established groups that were already forming, parent support groups and advocacy groups in the Fragile X field, and we realized that there really wasn't a whole lot of research going on about Fragile X. Fortunately, at that stage of the game, the gene had been discovered, because the reason they call it Fragile X is that the X chromosome appears to have a fragile site on it. It actually appears to be bent or broken. Only when it's incubated under special conditions, but it gives the name Fragile X to the condition, and it turns out that the gene is actually located at that site. So that really assisted in the Human Genome Project finding the Fragile X gene. So shortly after the gene was discovered, the scientists who were working on it were able to make a knockout mouse. So there was an animal model becoming available just as we were getting into the field. And of course, I'm a medical person. My wife is a computer scientist. And at that time, the internet was just becoming a major force in everyone's lives. And so when you're dealing with a rare disease, it's hard to get a lot of people together. But the internet allowed us to do that and to communicate with other families all around the world. So we decided we would start this organization, this nonprofit, to raise money for Fragile X research, especially with an eye toward developing new treatments for Fragile X, and fund biomedical research that would actually tackle this problem. Now, a lot of people thought that was kind of a crazy idea at the time because this is a form of intellectual disability, an autism spectrum disorder. And people thought, well, that doesn't seem like a treatable disease. That's just the way these kids are. But obviously, looking at the problem from the scientific level, this is clearly a single gene disease. It results in cellular abnormalities that are potentially treatable. And so we saw Fragile X right from the start as a treatable condition. And I think a lot of other people have come around to that point of view.
So what kind of research have you funded over the years? Has it been a lot of drug repurposing, putting things into clinical trials, or you mentioned that there's very little known? Did you all invest in some earlier stage research to try to understand the more fundamental biological nature of the disease?
Yeah, right from the beginning, we adopted this approach of doing biomedical research to look for the basic mechanisms of disease. And so, like a lot of rare disease foundations, we looked to academic labs to try to figure out what was going on. So we funded a lot of research initially on basic mechanisms of disease, but we also were thinking right from the beginning about more definitive therapies, things like gene therapy, gene replacement, gene editing, protein replacement. And in the case of Fragile X, we have the particular option of a gene which is fully functional, but it's been silenced. And so we actually have the possibility of doing gene reactivation. A lot of those technologies looked really promising circa 1994, but I think it's safe to say that at that time, the technology just wasn't really ready for prime time. So in the end, we started doing more and more research that was a kind of rational drug discovery process for Fragile X. And I think we were really successful with that, but we also began to appreciate the challenges of identifying treatment targets and then coming up with drugs that could actually make a difference in a human being, especially an adult human being with a developmental disability. So we had a lot of challenges, but a lot of successes, and we ended up having quite a few clinical trials that came out of that. Unfortunately, nothing has really popped yet. We haven't had the big success that we had hoped for by looking for investigational drugs that could treat the pathways involved in Fragile X.
So maybe this is a good segue to ask you to introduce yourself, Bruce. So Bruce was the founder of Cures Within Reach, which was a not-for-profit also focused around rare disorders and drug repurposing. So maybe we can try to weave these stories together shortly. But Bruce, I was wondering if you could just give us a quick background of how you got into rare disease research and advocacy, and what brought you to Healx today, where you're working on collaborating with not just groups like FRAXA, but many others.
I started in this space in 2002, and I got there in a very circuitous route. So when I was growing up, I wanted to be a dentist, and it was my life dream. So I went to dental school, and partway through dental school, I realized I had some vision issues that were going to make it really difficult to be a full-time dentist. This was a very long time ago, before they invented the microscope lenses that are now embedded in a lot of dentist glasses. And had that been around, I'd probably still be a dentist.
Right.
So I went to law school thinking I needed a new career. And in the late 80s, I came out of law school, and I got, because of now having a background that could be regulatory interested and also having a healthcare background, I started to slide into clinical research and regulatory affairs and professional relations in the healthcare research space. I also did a little bit of work in the professional liability, medical malpractice space, but I've been involved in medical research from back in the 1980s. And after spending 20 years in Fortune 500 companies, I decided I wanted to give back, and I became the executive director and later president and chief science officer of a family foundation. And while we were working as part of that family foundation, we, without much thought to it being drug repurposing, funded a number of drug repurposing clinical trials, which turned out to have spectacular results. So taking fairly innocuous drugs that were designed for disease A and repurposing them for disease B, and that turned out to be very powerful therapeutics. In several of these situations, they treated rare diseases of children that were at the time deadly, and were able to basically return these kids almost to normal health. And as we started doing that, we thought, this is really interesting. We're funding all this new chemical, new drug research and not getting anywhere with it. And we're funding these little pivotal clinical trials in the drug repurposing space and actually saving lives. So in 2005, we pivoted to be a drug repurposing nonprofit and started a public charity that's now called Cures Within Reach. So I was at the helm there for 17 years until I stepped down early this summer in 2019. And along the way, I had spent some time with Tim Gwillems and Dave Brown, the co-founders of Healx. I was on their science board. And when I was looking for my next adventure, I talked to them, and I connected with them fairly quickly after I stepped down at Cures Within Reach. Now, I know Mike and FRAXA for two reasons. So first of all, we followed FRAXA and were involved with them in our work at Cures Within Reach because they were doing repurposing with Healx and others. And also, I happen to have a family connection to this too. My nephew, Alex, who's 35 right now, also has Fragile X syndrome. And so I've been very involved in helping my sister-in-law and my nephew try and figure out what to do about it. We're lucky Alex is very high functioning with his Fragile X and doesn't have a lot of, some of the more severe symptoms, but we're still looking for therapies that can help him lead a more normal life.
So with the Healx collaboration, Mike, what were the kind of therapies that you originally started pursuing, and how did that process go? Did you get in touch with them? Did they get in touch with you? Did you meet at a conference of some sort? And how did that actually, where did you start and where is it at today?
Well, as usual, it's all about personal connections. And in this case, we had been working with a number of pharma companies of all different sizes, including some small startup companies. And actually, one of the small startup companies that we were working with was also based in Cambridge, England. And the CEO of the company, who I'd been in regular contact with because we were testing some of their drugs, some of their investigational drugs in the Fragile X animal models, he said, have you guys been interested much in repurposing available drugs? Because I know this guy, and the company's just down the street. They call it Healx. It's kind of a new company, but they do consulting on repurposing available drugs. And I really like these guys at this company called Sentinel Oncology in the UK, and we've been working with them for a while and kind of felt that they liked us too, and they had our best interests at heart. And I thought, well, maybe I should talk to this guy. So I talked to Tim Gwillems, and right off the bat, we could tell that we were seeing eye to eye, and we really had the same goals. And so we decided to do a repurposing project with Healx. And just from a research standpoint, it's very economical. It doesn't cost a lot of money to do this. So compared to what we would have spent on any given postdoctoral fellowship for a year, for less money than that, we were able to do this repurposing project. And after a few months of study, they came up with a list of repurposing predictions for us. And the really cool thing about that list, which had a kind of top 8, so the big 8 predictions, included 2 drugs that we were already working on, and they had no way of knowing that.
Right.
And then it had some other drugs that we hadn't been working on, but we had the ability to test these things in the animal models because we maintain that capacity in our organization. And I think that's really important. And so we immediately plugged those predictions into the wet lab, because remember, these are all artificial intelligence generated predictions coming from a computer and curated by the staff at Helix, where they have a lot of experts in pharmaceutical development, but nobody's actually testing these things. And so we decided to test some of them. And what we found was that some of them worked really well, some of them not so well. So we got a spectrum of response to these compounds, which I found reassuring because, frankly, there's this opinion going around that everything works in animal models and then nothing works in the human. But that's not the case. We have a lot of experience working with the animal models, and we definitely know when something's working and something's not working. So we found a couple of real winners that were not on our radar at all, unlike some of the other predictions which were on our radar. And so that was really exciting, that for less than the cost of a single postdoc in a single lab for 1 year, we all of a sudden had some new, not just treatment targets, but actual available drugs that we could put into clinical trials. And so that's what we're doing now.
Amazing. So Bruce, how does the process actually work? The machine learning or artificial intelligence approach, how do you spit out potential repurposable treatments?
The first part before the spitting out is how do you create the Helix platform? We call it HealNet. And it has 4 main components to it. The first one is what we call natural language processing. What that means is we gather all of the available scientific and medical literature, and we put it in a format that a computer can read it. And then we train the computer to read through as if it's reading natural language and make connections between words and phrases in a way that a human would. And the benefit of having the computer do this is that the computer can read millions of pages much faster than the human at light speed. And it can remember and document everything that it's read. And then it can make connections between what it's reading now and what it's reading 5 seconds later and 5 seconds later. And it builds up a database of how things are connected. And what's especially remarkable about this kind of machine learning, artificial intelligence is not only can it amass a huge amount of evidence, but it finds what would really be non-obvious to a human reader, because we just can't keep so much information in our brains at the same time. And the trail of getting from point A to point Z or point Z would be so complicated that no human would be able to follow it, or not many of us. But the computer can keep track of it. And then at the end, you can program the computer to display all of its information in rank order of why it thinks the top is more important than the bottom. And with a click, you can see the evidence chain that the computer used to generate why it thinks this is important. And then the humans, as Mike said, we have a crack staff of pharmacologists and other scientists who can then go and look at what the computer has decided and help to then figure out what the computer did really well and what it didn't do so well. Take what it did well and move it forward into, as Mike said, now we have some predictions at work, and then go back and retrain the computer that next time it doesn't make the same few mistakes that it made. That's the machine learning part. So the next time you run another query on the computer, you hopefully get a better set of results. And each time we do that, the results get a little bit better. And sometimes we'll go back 6 to 12 months later and rerun the same disease through the system, and we'll get even better results than we did the first time because we've retrained the computer each time we've run, and we're running these, several times a day and going back on a weekly basis and updating and doing the machine learning. So it's a really powerful way of helping humans to do a better job with the training that they've had.
Right. So it's constantly iterating and updating. So you have this web of knowledge that's the computer reading essentially all of medical literature, plus you bring in laboratory data and other kind of things and build this knowledge graph, right? I think I've heard you all call it a knowledge graph before, that allows you to make connections between existing drugs. And I think it's really important to underscore that we pretty much know they're already safe, which allows you to skip tons of steps in the process in terms of developing a new drug, right? So Mike, you received this list of 8. I think it's amazing that 2 of them you already knew about. It's a kind of internal validation that probably gave you much more confidence in this startup company at the time that no one had really heard of. And so where are you at today? You've been working on not just the mouse models, but I suppose next steps, thinking about whether any of them are viable for a clinical trial or pivotal study.
Yeah, definitely. In fact, we were so happy with the results from the first study that we did. We did a second study that was focusing on combinations of available drugs.
Right.
And the reason for that is, realistically, you have to assume when you're dealing with a major neurodevelopmental disorder, especially in somebody who's a little bit older, it's probably going to take more than one available drug to make a big difference in their condition. And you need to move the needle enough in clinical trials that you can really see a statistically significant effect. And one of the problems that's been really hard to overcome in any kind of CNS drug development has been that clinical trial stage, where you can often see some improvement, or you'll see improvements in subgroups of patients, but you don't get that overwhelming positive effect that you're looking for that says, yes, this is the treatment for this condition. And so we figured by going to combinations of available drugs, we could get a more powerful effect. And that's where this AI approach really pays off, because it can go through all the kind of multiplex variations involved in combinations of drugs and how to assemble these combinations intelligently. And then we started testing those combinations in the animal models, and now we're actually in the late planning stages of a clinical trial for combination therapies in Fragile X, which is really kind of a first. It's a difficult problem to tackle in clinical trials, because you can imagine that if you have 2 or 3 drugs and you need to test each one of those individually and each one of those in all the available combinations in humans, that becomes very time consuming and very expensive. And especially in rare diseases, it's you often don't have that many patients available to you.
Right.
So you have to be smart about how you do it.
Yeah, absolutely. And it's in rare disease, actually, like you said, just finding enough patients that fit the criteria. Sometimes the criteria for these trials are so specific, and they ask a lot of the families as well to multiple visits, sometimes traveling. We had a previous podcast episode where we talked a lot about this, that you have to really make sure the trial is set up right, because you often only have one shot at it. And if it doesn't go right, then it's many millions of dollars and a lot of time and hope in many cases as well, dashed.
Yeah, and I want to add a couple of things to what Mike said. One of the nice things about combinations is you could often then give the drugs in the combination at a lower dose than you might give if it was just a single drug. And so you can get synergy, where if you gave drug A, you'd see this effect, and if you gave drug B alone, you'd see this effect. And when you give them together, you actually see an effect that's more than what you would see just adding the 2 effects together. So often their mechanism of actions or targets or pathways that these drugs work on, where they actually help each other work better. So, that's the first thing. And the second piece is, and we're working with FRAXA on this, is to think of in these kinds of CNS diseases, are there ways to get more objective outcome measurements that would show that the drugs have done something powerful in the patient? Because often in these diseases, you're measuring things like how long does somebody make eye contact, and are they happier or less anxious? And it's a little more subjective and difficult. And I know, both for Andy with, Mike and his family, and also for my nephew Alex, they've both been involved in clinical trials where their families saw what they thought was pretty remarkable impact of some therapies that were being tested. But overall, it was hard to validate that they made enough clinical impact to get those drugs approved. And I know how devastating it was both for our family and for Mike's family when those drugs didn't move towards market, even though we thought they had made huge impact on our boys. And so as we're planning these clinical trials with FRAXA, we're also looking at, are there ways for us to take the knowledge that we have and find some more objective outcomes that, could help us measure things so that these therapies will actually get approval and can move to the patients.
Right. I actually read there's an article, I think it was in NPR, Mike, about your experience and your wife and Andy's experience in clinical trial where you felt that it was working, but in the aggregate, the trial didn't succeed for one reason or another. So it must be a very frustrating and in some ways motivating process to do better this time.
Definitely. We learned a lot of lessons during the course of these large-scale trials that have been done on some of the treatment approaches that we developed. And so we were pushing these things very hard because we thought for sure, in the particular case of the mGlu5 antagonists, we thought for sure these drugs will work in Fragile X, and they worked incredibly well in the animal models. And then when we got into the clinical trials, they worked incredibly well in our son. But then what we saw over the course of many months was a kind of habituation, a kind of tolerance developed to the drug. And I think that points out an important problem with investigational drugs in general, and an important advantage of repurposing available drugs, in that some of these investigational drugs have problems with them that you can't anticipate in advance. You don't really know how humans will respond to these drugs until you try them. And in this case, the drug worked great, but then we kind of lost the effect over time.
Right.
So that's a big problem with investigational drugs and repositioning, you might call it, these investigational drugs for a rare disease, which is what a lot of rare disease foundations do. A lot of rare disease foundations are doing academic research where they start out by identifying a treatment target, and they say, look, are there any drugs that can actually address this treatment target? And they say, yes, there's a drug in development with this pharmaceutical company over here. And the problem is that you can't get access to that drug if they're developing a blockbuster indication like Alzheimer's, for example, because they don't want to jeopardize that major drug development project for your teeny tiny little rare disease. And so you have to wait till those drugs fail for their primary indication. So one of the things I've mentioned before, I think, is that failure is almost a prerequisite when you have that approach. You're working with this pool of failed drugs that you know for sure don't work for a whole bunch of things, and you're hoping it'll work for your special thing. Whereas with repurposing available drugs, you have a pool of drugs that you know work. They're good drugs. They're already on the market, and they work for, in most cases, more than one thing. So it's really a big difference.
Yeah, I was just going to say it's fascinating as well how some of these drugs are so simple in what they do, and as you mentioned earlier, it's such a complex. Issues with the brain are so complex and happen over decades. It's kind of miraculous in a way that a relatively simple chemical could help. And I suppose this is where we get into the combinations where at age 35, it's very unlikely that there's a silver bullet where you take a pre-approved drug for something else and it just happens to work. But potentially by really tweaking dosage and different combinations, you can start to get closer.
Yeah, and it's impressive when you actually look at some of the long-term results in populations where available drugs can make a big difference in diseases that are difficult to treat. There was a recent paper that was basically a study of the Swedish National Healthcare Registry, where they looked at all the records for everybody in Sweden over, I think, a 15-year period. And they looked at the records for people with diagnoses of schizophrenia and bipolar disorder, which are really tough to treat conditions, chronic mental illness, severe mental illness. And they identified 3 classes of compounds that were effective in decreasing rates of hospitalization in those patients, psychiatric hospitalization, so non-psychiatric meds. And they found things like statins, calcium channel blockers, and biguanides like metformin. So some of the really popular repurposing candidates that have been mentioned for a lot of things, including Fragile X. The thing is, in psychiatry, we identified calcium channel blockers a long time ago as being potentially useful in these conditions. And they've done many rigorous clinical trials with calcium channel blockers, and they always come up with mixed results, just using this one drug by itself. Right. So ironically, if you look at thousands of patients over dozens of years, you can see very clear beneficial effects from these drugs. But when you actually do a traditional clinical trial, you lose that effect. Right. It just gets lost in the noise.
Do you think, Bruce or Mike, that that repurposing is a solution to the potential high price problem in rare disease? So every year there are many more transformative drugs coming out using RNA interference or exon skipping or other kinds of next generation therapies, but almost without exception, these carry price tags of hundreds of thousands or even millions of dollars per patient per year in some cases. Is repurposing potentially a solution to this problem as well?
Well, we hope that it is for any number of reasons. So first of all, if you can reduce the timeline to get a drug to market, and you can reduce the cost of the clinical trials and the preclinical development, then you have less investment that you have to recoup over the period of time. So to start with, it should be cheaper from that standpoint. The second thing that makes it less expensive is most of the drugs we're going to repurpose are generic drugs that already are widely manufactured, and we know how to make these. So the cost of goods is fairly low as compared to making a monoclonal antibody or something that has to, incubate over a long period of time. So the costs are low. And, obviously every commercial company needs to be commercially viable, so we can't do it for free. But part of the reason I joined Helix over the summer was the commitment of the founders, the board, and the investors that in every way possible, when we can bring a drug to market, we want to bring it at an affordable price. And we hired a company to look at, for example, the Fragile X market. And we said, if we were a company that was trying to maximize profits, what is the maximum price we could bring out of this combination of repurposed drugs? And then at the other end, we said, so if you look at how much investment it's going to take for us to get this drug to market, what's our break even? And then we said to them, what's a reasonable price that's closer to break even than it is to maximum available price that provides enough profit for us so we can continue to do this kind of work and is still reasonable for patients and payers? And it was interesting for a disease the size of Fragile X, with the kind of penetration we think we'd have, the percentage of patients who might have good use of the particular drug. And we can bring it out, for many less zeros than, the ones that you were talking about. I think it's amazing the breakthroughs that gene therapies and antisense oligonucleotides and other kinds of things are eventually going to bring. And obviously over long periods of time, we'll figure out how to do that more cheaply, I hope, because we won't be able to afford it economically. We're unable to fund every rare disease at $1 million a patient. So we're very hopeful that we'll, where we have control over the drug price, that we'll be able to bring these out at very affordable levels.
Yeah, and I would also like to point out that it's not an either or proposition. It's not that we have repurposing with available drugs or we have definitive therapeutics like gene therapy. We can have both.
Right.
And I think we've seen from the cases where gene therapy has become a reality, from those rare exceptions, like, for example, everyone is talking about SMA, spinal muscular atrophy, and the fact that there's a gene therapy for it. Well, that's great, but it has to be done very early in life or it just doesn't work. So there may be similar developments in Fragile X that are developed, which are effective early in life. But if you give them to people who are older than 3 or 4 years old, it just doesn't work. That's where I think you still need to have better therapeutics for the folks who are around and alive with Fragile X, because they have a normal life expectancy. They live a long time. There's a lot of older people around with Fragile X.
Yeah, it's a very good point. And I suppose also you can imagine a world where there's a frontline therapy that we know works for the majority of people, and develop some way to determine who that is going to work for. And if it's not going to work for them, then you move them on to something potentially more expensive, in the way that is done with cancer, where they don't go for the latest breakthrough drug on everyone. They try what they know works, and then only shift to the more expensive things when it has to be done. Because as a society, I think if you were to do the math on 7,000 rare disorders and 1 in 15 people affected by a rare disorder, if we're spending $1 million a year per patient, it's just not going to work. But in the cases where it is truly life-saving, then we have to find a way to make it work, right?
Absolutely. Yeah. And, the other thing that we think about, Patrick, is that we're not just doing this for developed nations. There are people with Fragile X all over the world, and we want to make sure that, in places where nobody could afford $1 million therapy, there might still be an affordable therapy that patients can manage. The other thing about repurposing that I think is incredibly powerful is when you have a success in repurposing, it often helps you see what other kinds of things might be helpful, whether it's a new drug that could be developed that could be even better than the repurposed drugs, or additional repurposing that could be helpful. When I was at Cures Within Reach, one of the projects that we had invested in that was taking place in Germany actually has a 9-drug repurposing regimen in addition to the standard of care anti-cancer drug in recurrent brain tumors. And it took 4 or 5 years for a group of people using AI and other resources to come up with these 9 drugs, none of which are oncology drugs, but they all do something in cells that doesn't overlap with something else and doesn't thwart what one of the other drugs does, and in order to begin to help the cancer drug work better and help the patient's own immune system and other mechanisms work better. And my guess is that as we get more advanced in our work, both with AI and precision medicine and with our understanding about drug repurposing, diseases like Fragile X that have so many components to them could be amenable to an even bigger cocktail of low-dose drugs and even nutraceuticals, which are drugs that have never just been approved as drugs.
Right.
There's a lot of nutraceuticals out there that have very powerful biological activity in the body, and they just never been approved as drugs.
Right.
And if we can get them approved, maybe we can turn them into drugs so that you get the same dose and the same kind of quality that you get in drugs. But we know that they they're helpful in patients and safe to use.
So,
yeah, I think we've been asking the question the wrong way for many years, and we start to think about these drugs that are highly targeted interventions that affect a specific pathway, and that comes from academic research that's very narrowly focused on something going on in the brain, for example. And then you try to get a drug that addresses that pathway, and you try to take that through clinical trial, get it approved. That's one way of doing things, but it hasn't really worked out. Maybe a better question is, let's say we've got a pool of available drugs, which are, say, the usual number people float is around 2,000 unique chemical entities that are available, but really it's just a couple of hundred drugs that are suitable for administration to children, for example, and things that get into the brain. So you've got this relatively small pool of drugs. Maybe the real question ought to be, what are the best 3 drugs for people with Fragile X to be on long term for optimal functioning and quality of life, kind of like that Swedish study. What are the best 3 drugs over a long period of time that would work out for folks? I always think of these studies that show that places that have lithium in the groundwater, even though it's in very small concentrations, they have a much lower rate of suicide and homicide and accidents and depression and all kinds of things. And it's something that you can only see over the long term. But I think what we have now is a technology that may be able to get a handle on that. And once you get a handle on that for one condition, you can start to train the machine, look at all kinds of other conditions, because a lot of these things are related. Right.
Yeah, I mean, I think that's a perfect segue to the question I was going to ask Bruce is how does this scale? How do you go from, my understanding is FRAXA is one of the first really successful programs that you've run, but the plan now is to do this dozens or hundreds of times. How does that play out?
Well, just what Mike was sort of talking about, thinking of what group of drugs could be used in one disease. We also think about what disease issues are present in multiple diseases. What kind of disease clusters show similar phenotypes, signs, and symptoms that Fragile X has, where we might be able to utilize the same technology, maybe the same drugs, to find repurposed therapies in those. We know from our experience at Cures Within Reach, we were involved with a lab and a physician scientist at Children's Hospital of Philadelphia who found a drug that could be repurposed for what's called a pediatric autoimmune cytopenia, which basically means there were some white blood cells that were not performing as they should in the body. And because of this poor performance of these white blood cells, they would build up in the body and crowd out the healthy spaces, and the kids would eventually die. We repurposed this drug for that disease, and when it was successful, 85% of the kids were in complete remission in 90 days on a single dose in the morning and the evening. And it's now been since through 2004. So in 15 years, no dose escalation and no acquired resistance. These kids are doing great. But we went back to the researcher and we found 6 other similar diseases, and we did a clinical trial in those. And in 4 of those diseases, the same dose of the same drug rescued those patients in those similar but different diseases. And so we really feel like there's an opportunity for drug repurposing. If we can create the success we believe we'll have in Fragile X, we know there are other rare diseases that, show those same phenotypes, the same signs and symptoms. The other thing that's obviously interesting, and Mike sort of alluded to it with the low dose lithium in the groundwater, but some of these repurposed drugs are also going to be useful in more common diseases, but it's easier to get them approved and get funding to test them in the rare disease. And some of these drugs, and there's lots of people doing work in this now, might be important for just general health, improving the quality of life if it doesn't improve the length of life. But some of them, like rapamycin and metformin, have been tested in laboratory animals and have shown that you can increase quality and length of life with low doses of these. And there are some studies like the one Mike referenced in the lithium population, where you can see patients who are on long-term of a particular drug. Not only did it work for whatever condition like type 2 diabetes, but it helped in other things in their general health. And so we strongly believe in the idea of using the medicines that are already available, because we know how they work and we know how they're safe and we know their interactions with other drugs and other diseases. So eventually, my guess is that, precision medicine is going to allow us to do diagnosis far ahead of how we can create new on-the-spot therapies. And so the best we'll have is the drugs and the nutraceuticals that are already available.
Right.
And, 10 years from now, there will probably be tests where you can go into your doctor who will say, hey, based on what's going on in your genes right now and in your proteins and in your blood, we suggest you go on this cocktail of low dose existing drugs, because we think that'll help. Not just, a drug for cholesterol, but a drug for overall health, because these things are complicated and interact with each other.
Right. I love that vision for the future. That'd be amazing. How
it's only possible with available drugs. If you think about it, you can't just go and make a new drug for every condition, because we know how difficult that is, and we know what the failure rate is, and it's 99+%.
Right. Yeah. And the expense just doesn't add up, right? I think it costs upwards of $1 billion now to get a drug to market. So as our understanding of disorders increases and we split them into more and more precise groups, the math is just not going to add up.
Right.
And what's interesting, I think, is combinations of existing drugs really create in the body what you might get by developing a new drug, but even better because they'll hit multiple targets at the same time. It would be hard to create a drug that would do what a combination of two drugs in Fragile X will do. It would be too complicated to figure out how you create that kind of a drug.
Right.
So it's really a very powerful way of thinking about it. And what's interesting is, over the last 15 years or so, as I've been working in this space, we've watched drug repurposing go from unknown in 2005 to sort of disdain in the late 2000s. Everybody turned their nose up at it, to being interesting in the early 2010 to 2015 and being kind of all the rage right now. Every pharmaceutical company has divisions that look at their own assets and see what they can be repurposed for. And so I think it's going to continue to gain popularity, and it's going to be essential if we're going to improve care and reduce cost, which is the holy grail of healthcare, right?
Right.
Right now, new chemical entities, new medicines improve health and increase costs, and that's unsustainable. So hopefully repurposing can be a part of bringing the costs down and improving the care.
Mike, what does...
the more we do it, the better we'll get at it. So, when Healx said that they want to find repurposing solutions for 100 rare diseases over the next 5 or 6 years, that may be ambitious, but that's not unrealistic, because the fact of the matter is, once you get the first 10 or 15, you reach a tipping point where the knowledge in the system becomes so great that the next few become a lot easier. And then after that, the next few become really easy. And at a certain point, the next few will become trivial.
What, Mike, does a patient organization or a foundation need to start a program like this? Do you need to have some connections to academic lab or your own academic or your own wet lab to do the follow-on research, or what does if someone who's listening who's part of a research foundation or patient organization, how do you recommend getting started on something like this?
Well, I consult with a lot of different rare disease foundations, and most of them are a lot younger than FRAXA. because most rare diseases have been more recently recognized than Fragile X. I do think it's important to have the capability to do the lab work, but I would also say that over the years, one of the most important lessons that we've learned is that you can use contractors, you can use CROs. They don't have to be academic labs. They're out there. There are people who can do this work for you for a reasonable fee and with complete accountability. So you pay them the money, they give you the results. And that's something you don't always get in academia. So I think the academic model is part of the problem, and it's part of the system that doesn't really work very well. It's very inefficient. And especially as more and more rare disease foundations pour into that space and try to get academic labs working on their rare disease, you're just kind of bidding up the cost to do the same thing. It's making it more and more difficult. So I think using contractors to do what we've done through basically a FRAXA internal resource, I think is a viable possibility. But obviously, it can't all be done on the computer. You have to have clinics, you have to have labs, you have to have animal models or cell models. You have to have something.
And Bruce, I guess with the new rare treatment accelerator that you all are working on, Healx now has a lot more money to work with because of some of the early successes and the belief from the investor community that this is a way to change treatment in rare disease. So you have a program where patient organizations can speak to Healx, and Healx will provide, I think, up to $1 million or pounds. I'm not sure, of funding to do some of this early development work. Is that right?
Yeah, we have that program, the Rare Treatment Accelerator program. We announced it in late November, and the first applications are due in January. And the idea there, and sort of to answer, in addition to some of the things Mike said, we're looking for patient groups that have deep disease knowledge, that have access to networks, cellular models, animal models. They don't have to own any of them themselves, but they have to know where they exist and which ones make sense. They have to have patient knowledge so that we can work with them to say what's the most important thing for us to go after if we're looking for therapeutic value for your patient population. It's interesting. I'll give you a quick example. So there's a rare disease called Prader-Willi syndrome. And if you look at it, you might think intellectual disability or some growth issues might be the things that the parents and the patients would be most concerned about. But when we talked to the patient group and the parents, we found out the number one thing they want us to deal with is sleeping, because these kids don't sleep through the night. And if they don't sleep through the night, the parents don't sleep through the night. And if everybody's exhausted, the rest of it doesn't make any difference. And we would never have found that out by going to the literature. There's no publication that says the biggest issue in Prader-Willi is that kids don't sleep. But the patient group brought that kind of knowledge. And Mike and the people we've dealt with at FRAXA have told us all sorts of things that we would never have been able to get from reading the literature, talking to the scientists, talking to the clinicians. That's so incredibly valuable. And because of that, we're able to both focus our work and speed along the development. I mean, we basically went from starting the predictions in Fragile X to being in a position to write the clinical trial protocol in 18 months, which, is like light speed in drug development. And, as we continue to do this and get better at figuring out how to get these clinical protocols written quickly and move them along, we're hopeful to be in the clinic in less than 24 months in most of the diseases. And we do have $20 million or 20 million pounds. It's close these days. So, unfortunately, but we don't have a set amount that we'll commit to a particular disease. We'll do what's necessary to get the predictions made and the preclinical validation complete. And in some cases, it'll be less than $1 million. In some, it'll be more. But as long as it's moving towards looking like it could have a clinical impact on patients, we'll keep funding it until we're at the point where we can either decide there's no value there or we're ready to move into the clinic. And at that point, we'll have to raise more money because the clinical trials are more expensive. But we should have the assets, the commercial viability, and the patient support in order to raise those funds.
Great. No, that's very exciting. So when we put the episode out, we'll make sure we remind anyone who's at a patient organization to take a look at that and see if it applies to them, because I do think it's impossible to do this without collaboration. When we had Nick Serow from the AKU Society on a previous episode, and they've recently had very good news about a treatment that they've been working on for years. And the takeaway was you have to have dedicated patients, dedicated researchers, dedicated commercial entity that's going to help with the clinical trial or bringing the assets. So I do think the name of the game is collaboration on this. There's no way for anyone to do it by themselves.
Right. And when this podcast is released, the nice thing about the Rare Treatment Accelerator is that we'll continue to have more opportunities for applying and decision making. So this is just our first cohort that ends on January 10th, 2020. But shortly thereafter, we'll have another call for applications, because our goal is to find at least 20 of these patient groups that we can work with in a serial fashion. So it'll take us 6 to 8 weeks to get each one started so that the predictions are moving along and we can move to the next spot, and then we can enter a new one in. And we're just planning on hopefully doing 2 to 4 of these every quarter for the next couple of years until, we have an entire pipeline of potential therapies moving towards the patients.
Great. That's really wonderful. Well, I know we've blasted through our hour of conversation. So I just want to say thank you to both of you for taking the time to do this. I think it's important it's so challenging to develop new treatments in rare disease. And when there is the rare exception where you get 2 groups or multiple groups that come together and are moving towards success, I think it's really good to talk about how that came about and how we can learn something more. And I really didn't expect the insight around how this can transform into health and wellness in general, micro-dosing of small, different combinations of existing drugs. I think it's a really fascinating view of how the future could be. And I think it makes sense in a lot of ways and solves some of these problems. So thank you both for taking the time.
Well, it was my pleasure from Healx. And I just want to say, Patrick, really appreciate you taking the time to highlight all of these issues on the podcast, and you do a great job on that. And I also wanted to say how honored we are to be able to work with Mike and FRAXA. I've known Mike for a long time. He's an amazing individual, and he sets the bar pretty high for other patient groups. For us, we come with very high expectations about what patient groups can bring to a collaboration, because Mike was the first one. It's kind of like drafting Michael Jordan as the first player on your basketball team. After that, it makes it hard to think you want anybody else. But and Mike has been very gracious about talking to other patient groups about how Healx works and others. So, Mike, while I have the chance to say thanks to you and, how honored we are to work with you, and how we're also really honored to be moving some treatments through to patients. It's what gets us up in the morning and what we think about when we go to sleep at night here at Healx. Obviously, we're a small commercial biotech, but what really drives all of us is the thought that one day there might be a miracle for patients. And if we can be a small part of that, we're happy to do it.
Well, I really appreciate that. And we really appreciate the chance to talk about Fragile X and to get the word out. And Healx has been great to work with. And I'm not afraid to tell anybody about those experiences. It's really just been a complete paradigm shift. And we really think it's the way to go.
Great. Well, thank you guys. This is one of the favorite parts of my day because I get to just hear from really fascinating and smart people talk about things that I'm interested in. So it's really my pleasure. We'll keep track of the progress from FRAXA's side. I'm sure it'll be in the next probably 12 to 24 months. We'll be looking at clinical trial. And then on the Healx side, I think the Rare Treatment Accelerator is a really exciting new development. So hopefully we can keep track of you all and some of the first patient organization that you start to work with, because I think it's a very cool and scalable model that can hopefully make a big difference in rare disease.
Thanks, Patrick.
Great. Thank you both.
Thanks a lot.
Bye-bye.
Bye-bye.
Bye.